Mitosis is a type of cell division which occurs mainly in somatic cells and stem cells. Now, this mitosis also occurs in regeneration of the organs. We have seen in regeneration occurs in planaria, in lizard. So there also mitosis will occur for extension of the body parts. Now, during this process, basically there is a separation of nuclear chromosomes into two identical daughter nuclei. It occurs in two stages, karyokinesis, and the other one is cytokinesis. Karyon means nucleus. Kinesis means breaking. So breaking of the nucleus. And the other is cytokinesis. Again, it means breaking of cytoplasm into two parts. So now this karyokinesis is completed in four steps: prophase, metaphase, anaphase, and telophase. Before the cell enters into the phase of prophase, the cell undergoes a period of growth and rest. Here, the genetic material of the cell increases. Now, during prophase, the chromatin material condenses and becomes short and thick to become chromosomes. Each chromosome has a sister chromatid joined at the center by a centromere. So now here we can see this centromere. This is the centromere. Centromere is in mid of the chromosome. Now here the nucleolus and nucleolus disappears and the nuclear membrane starts to break down. Centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Now, during metaphase, microtubules of the spindle interact with chromosomes, and this causes chromosomes to move at the equatorial plate. They are moving at the center of the cell. This is the equatorial plate. Now, special type of flexible protein fibers, that is spindle fibers, are formed. These long thread-like structures you can see form between centromere of each chromosome and both centrioles. Now, after prophase, we have metaphase, and after metaphase is followed by anaphase. During anaphase, sister chromatids separate at the kinetochores. Now, these two chromatids are joined to each other by kinetochore. And now they separate at the kinetochore, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles. They have moved. Now they were in between at the equatorial plate during metaphase. Now they are moving towards the opposite poles. As the microtubule shortens, these fibers shorten, they contract, so they are moved upwards. Here, the separated chromatids are called as daughter chromosomes. Now, here onwards, we are going to mention them as daughter chromosomes. In this way, each set of chromosome reach at two opposite poles of the cell. Now, during anaphase, they have moved at the opposite poles of the cell. Anaphase is completed at this stage. Once they have moved to opposite poles, the anaphase is completed. And the last phase is telophase. Last stage of karyokinesis is telophase. The chromosomes which have reached at the opposite poles of the cell now start to decondense and decoil, decondense and decoil. So they will be, which again which again become thread-like, thin and invisible. Nuclear membrane. We had seen nuclear membrane had disappeared. Now this nuclear membrane is formed around each set of chromosome reached at poles. Nuclei are also formed. Nucleolus also appears in each daughter nuclei. Spindle fibers completely disappear. So here there is reformation of nuclei along with the nucleus. So we have seen at the end of prophase, the nucleolus and nuclei, they had disappeared. But now there is reformation of nuclei along with the nucleolus. Now the last phase is cytokinesis. Second stage is cytokinesis. It is nothing but the partition of the cytoplasm. So here cytoplasm divides into two new cells which are formed. Now, in animal cell, a furrow or a notch appears in plasma membrane, which deepens and divides into two daughter cells, each containing a nucleus. In plant cell, the cell wall formation is triggered at the center of the cell. This is the cell wall formation by the formation of cell plate, resulting into two daughter cells. So in short, we can say about karyokinesis, prophase, during prophase, the chromatin condenses and becomes short and thick to form chromosomes. Metaphase, microtubules of the spindle interact with the chromosomes and they come at the equatorial plate. Anaphase, the sister chromatids separate out at the centromere and move towards opposite poles as the microtubules become shortened. Telophase, the last phase of karyokinesis, daughter chromosomes arrive at the poles and the chromatins start to decoil, followed by formation of nucleus with nucleolus. So here now two identical daughter cells are formed in this mitosis.